Hello everyone, my name is Sagar Sharma and I will be presenting my final year dissertation project. It was on development of novel biomolecule cocktail for heavy metal stress tolerance in plants. We were supervised by Dr. Indu Bhatt, our professor at IMS Engineering College, and this is the project of year 2025-2026. So we are thankful for our guide, Dr. Indu Bhatt, and our HOD sir, Dr. Ajay Kumar Sharma, as well as our coordinator. Contents. So we'll be going through introduction, project goals, methodology, procedure, analysis, discussion, references. So when we talk about phytoremediation and mainly phytoremediation, genetic engineering or soil amendments, so we face the limitations in terms of time, cost, ecological feasibility, and regulatory acceptance or compliances by the government. So what we came up was to actually naturally externally supply nutrients or natural biomolecules that work in a way where plants' defense system is complemented. What leads to this complementation? What is the result of it is that the stress caused by the reactive oxygen species. The reactive oxygen species is caused by the stress, and these stress can be abiotic or biotic. Now, these reactive oxygen species damage different organelles and different parts, DNA, RNA of the cell. So, what plant does is produce antioxidants and other ways to reduce the damage. Now, at a point or eventually, the stress builds up and leads to reduced output, reduced food production, reduced uh, uh, life, lifespan of the plant. So, uh, what we prepared was giving synergistic biomolecule cocktail, which can be introduced in either the leaves or through the roots using water. So this biomolecule cocktail can be made up of essential hormones, amino acids, chelators, inducers, and signaling molecules. So hormones will be like auxins, gibberellic acids, cytokinins, ethylene, and whereas amino acids can be proline, etc., etc. So, so our project goal was to design to design the biomolecular cocktails based on how our plant was reacting to how our plant was acting against stress, and then study the effect of the cocktail which we gave stress to. So for making cocktail, what we did was we identified certain plant hormones, amino acids, and antioxidants from literature review, and studied the parameters on mainly three things: weight, root, and shoot, growth. We then performed a protein estimation to get an idea how the stress is working and how our cocktail is working. So concentration and decomposition of Hogland media. So we used Hogland media and used mainly hydroponics based system. So in Hogland media, macronutrients were potassium nitrate, magnesium sulfate, septahydrate, and all the main basic Hogland media components. So it was divided into macro and micro. Along with this, we gave copper chloride as stress. So copper chloride of different concentration, which we'll be going through ahead. So methodology, what we did was we took chickpea plant variety NC9, and NC9 was received from ICMR Delhi, which was again imported from Kanpur, IIT Kanpur, I think, ICAR Kanpur, sorry. So IARI Delhi, we received the seeds, and it came from ICR Kanpur, and the plants were grown in hydroponic system. The collected seeds were grown in Hogland medium, and different copper stress concentration were given. So in our case, heavy metal stress was of copper. The concentration were 0, 50, 75, 100, 125, 150, 175, 200, and 250 micromole. Main reason to choose all these concentrations uh, were to differentiate the stress into mild, mild, severe, and moderate. After that, we checked the effect of stress, heavy metal stress, using pigment analysis and protein estimation technique. Then, after identifying the mild, moderate, and severe stress ranges of copper chloride, we developed the molecule, and the cocktail. We tested our cocktail. So the seeds were soaked. Seed was soaked in petri plates with using the tissue, the blotting paper, for a few days. Then after the germination, these germinated seeds, after developing a certain length, which is of around 2 centimeter, 3 centimeters, we transferred it to these cups. So these cups had plants. So we used mesh to stay the seeds upright. We gave the photo period of 8 hours light and 16 hours dark. Transferred seeds were allowed to grow in hogland media first for a few days until both shoot and root is developed significantly. So first there was a root growth in germination, and then there was shoot growth as seen in figure 4. Now the analysis, the copper, as you can see in the picture, the copper stress, the amount of copper chloride concentration, we can see there is reduced growth over the shoot. Why is it not visible such goodly in the root cases? Because we allow a certain length to increase. So we allow certain length of shoot to root to grow, and then after that the stress reduces the growth rate. So our growth analysis was based on three things: root length, shoot length, and weight. So with over various concentration of copper, we came up with protein estimation. So we can see that there is a peak around 100 micromole, and we are so yeah. So at around 100 micromole, we have a peak of protein in shoot. This um, points towards the defense system of plant, that it is able to cope up with this amount of stress. It is moderate to it, but after that it wasn't able to keep up with the increasing copper stress and finally gave up. Seen similar type of graph was observed in root as well. However, the graph peak shifted more towards 125. So this is our pigment analysis. So as you can see, the pigment is reducing. So at zero concentration, our pigment was maximum, but as the stress increased, the pigment reduced drastically. So cocktail comparison. So in the left one we have cocktail A. So the very left one, for each photo, the very left one is 0 micromolar copper chloride, and then the copper chloride of A and B. So this is our cocktail composition. So cocktail A had citric acid, whereas cocktail B had more ascorbic acid. Proline were of similar. Proline in cocktail A was more, and cocktail B had less proline. Salicylic acid was less in 0.4 millimolar, and salicylic acid was 3 millimolar in cocktail B. We chose this based on the literature review and a previous study by our guide and her students. So cocktail. So we can see that as we are increasing concentrations, the plant size is reduced basically because growth is less. In first picture, there is also due to the clicking picture on the chart paper, there is some colored on the root. So somewhere it might look that the zero concentration or control has very less length root length, but it is actually longer if we see carefully. So in cocktail, when increasing the concentration, cocktail B had the growth better than the cocktail A, and that both their cocktail A and B growth were better than the control. So cocktail analysis. So we saw that cocktail 2 or cocktail B had the most growth against copper, increasing copper stress. The mean concentrations were 0, 50, 100, 150 and 200 microliter. And this is the graph you can observe. So control went downwards as the increase in concentration, but there is a peak which we can observe around 50 micromolar of copper chloride stress. The cocktails were given along with the copper chloride stress. So both the chemicals, both the solutions were added right when we added the stress. So Hogland media, then after a few days when the shoot length is significant enough, 1 to 2 days, grown the we added the stress and cocktail. Now this cocktail Now the cocktail with the study was for 6 days a week. So the values were taken to be from 0th day, 3rd day, 6th day. So we observed an increase and then decrease in protein concentration in root shoot as the copper's concentration increased. This is in the case of when no cocktail was given. Pigment in shoot decreased, whereas with increase in copper concentration. Cocktails were successfully synthesized and we were able to see its effect. Both cocktails showed peak mitigation at 50 micromolar concentration, and both cocktail showed increased relative growth to control even at high heavy metal stress concentration 150 and 200 as compared to the control one. However, the cocktail B had more effect than cocktail A. So this was the discussion and this project was successful. Our seniors also worked on mercury. However, the cocktail was not used. I think this cocktail can have many implications, and the mode of applying this is very easy. Uh, Through leaves, it can be sprayed on the leaves, or it can also be given through the roots. And uh, providing a concentrated solution to these cocktails and then diluting it at the site of usage is very easy, and it can be very profitable as well as um, very worthy of using at the places where factories in the village area or agricultural land is used, because mostly heavy metal senses is found there, and can also be used in reclaiming the already stressed area or contaminated area. So this project has many possibilities to work on ahead. So these are our references which we used earlier, and uh, thank you. Thank you.